Welcome to the last in-person workshop for, instructor-led workshop for our medical microbiology and infectious diseases bioinformatics workshop series. Today, the workshop is called Introduction to Tree Visualization and Annotation using ggtree, instructed by myself, Taylor Davedow. I'm a Ph.D. student in the Department of Medical Microbiology and Infectious Diseases at the University of Manitoba. So just a reminder that today's workshop is being recorded and posted to our MMID Bioinformatics Workshop YouTube channel. So if you ever want to go back and look at it and follow along again, it'll be available for you to do so. For live question and answer, you can go to Slido and use the participant code for today's workshop. As I mentioned, today is the last instructor-led workshop, but there are 2 additional dates available. If you would like to bring your own data set and sign up for in-person classes, then we'll be able to help work through any issues that you're having, any questions. In person, it'll be a lot easier. Otherwise, we can do it over Zoom, we will change it from in person. So sign up if you're interested in coming in person. Or anyone in person who needs to set up Wi-Fi. Are you guys all good? Yeah, experts by now. So our learning objectives for today's workshop is to create a tree using ggtree. We'll use geometric functions and aesthetic mappings to annotate the tree. And then we'll learn how to customize legends and add themes. The files for today's workshop include a tree file that's in NUIC format. This is what we created if you were here for last week's workshop by running IQtree. If not, it's still available to download. There is a metadata file in Excel format, and it includes compiled information from the manuscript we're working from. And then there's 2 R scripts available. There is one main script that you can open up and follow along for today's workshop, so you don't have to frantically type. It's kind of a lot to type for today. And then there's a second R script. Don't look at it yet. It is the answers to one of the exercises we're going to work through. So if you can open up and save the materials to your workshop directory, it's on our GitHub page for today's workshop. I can give you a few moments to do so if you guys haven't done that already. Let me know if there's any issues. Maybe just give a thumbs up or write in the chat when you guys have got that all downloaded, and then we'll move on. We're good? Yeah, okay. Okay, so we'll start off with getting started. So you can open up your RStudio. We're going to set our working directory to our workshop folder. So for this part, I will show you guys. Here. So my workshop directory is just called workshop_ggtree. And to set it as your working directory, you can go to this More tab, go down, and set as working directory. Once you do that, you can create 4 folders inside your working directory. I usually just do that in the the File Explorer here, and just create data, docs, output, and script. In the data folder, I will move in my metadata Excel file and my tree file. And this just helps keep things a little bit organized, especially when you're working with a lot of different data. Docs, I don't have anything in there right now, but typically I would keep like today's slides or something like that, like maybe the paper for reference. Output is going to be any output we can forward to that folder, and then script will have today's script. So move all the files into their appropriate documents. And then we're going to install the packages. So if you don't have any of these packages installed yet, in the script, you can just unhash them and install them. I already have them installed, so I'm not going to do that. You need to have this BioC manager package installed and then loaded. So once it's installed, you can load the BioC manager library, and this is just required to install ggtree. If anyone hasn't installed ggtree after last week, it might just take a few moments to do so. And while those are installing, I'll just move back to the notes. Okay, so we are working with the same publicly available data set from this paper, clustering of Vibrio parahemolyticus isolates using MLST and whole genome phylogenetics and protein motif fingerprinting. So the objectives of this study that the authors wanted to do was to cluster strains from clinical and environmental isolates of Vibrio parahemolyticus. And we only took a subset of samples. So I think 15 samples we worked with last time, including this large cluster here. And we ran them on IQtree. So just a reminder of the basic workflow, general data quality assessment that we had gone through in some of the earlier workshops. We've done taxonomic classification in the March 30 workshop, if you want to take a look at that. Genome assembly as well is part of a 2022 coding workshop, as well as some downstream analysis. So last week we did phylogeny using IQtree, and then today we're doing the visualization portion using ggtree. So ggtree is a really great package for our programming language under the Bioconductor project. That's why we have to download it through there. And it was created by Dr. Yu. And it's a great extension of ggplot2. So it's a tool that we can use to basically visualize and analyze and annotate phylogenetic trees along with their associated metadata. And it's really great because there are other packages or there's other, I guess, tools that we had looked at last week that are online, but what's the great thing about ggtree is that it's completely customizable and programmable. So you can have a custom tree available that is reproducible as well. So for this part, you can follow along in your R script. Is everybody's packages downloaded at this point? Once your packages are downloaded, you'll be able to load them. So to load them, we'll do library and then with a package in brackets. The read Excel file or package we will use for reading in the Excel file. The ggtree obviously will be for building our phylogenetic tree. There's tree.io package, which is helpful for read.newick function, which is used to read the tree file. PhyTools is another accessory tool that I've used to midpoint root a tree. So it kind of just roots the tree at the midpoint, you'll see. And it also can utilize the read.newick option. And then there's tidyverse, which is really great. Will assist us for tidying up any of the data if need be. And then ggplot2 should be automatically loaded in with ggtree. If you're having any issues, then it might be just make sure that ggplot2 is loaded. Does everyone have their packages loaded and ready to go? Yeah, okay, great. So the first thing we're going to do, so you guys, I'm not going to open up the R script because it's pretty much the same as in here. You guys can follow along. So under loading in the files, you can load in the tree and the metadata by highlighting them and clicking run. And that should help them come up in the top right pane as objects. After that, you should be able to create a basic tree. So to do that, you have to just, it's ggtree, and then the philo object in brackets. And our philo object from the last slide is our tree file, which we loaded in using read.newick. Once you load that, it should pop up in your plots. And it should be this just basic looking line tree. Was everyone able to get that? And if you want to look up any additional arguments, you can do go to the help page by doing !ggtree. You can also change the tree layout. So depending on your data and what you're interested in visualizing, you can change the tree layout by using the layout argument. So you can set the layout to rectangular, and I'll let you guys play around with that. I think it's in the code where you can run it as rectangular. You can try running it as circular. And then another one I put in there was, I think it's just like a rounded rectangle. But the rectangular is actually the default. So even if you don't set layout, it'll show up as rectangular. Other options that change the layout of the tree, if you change the branch length argument and set it to none, it'll actually make a cladogram, which will show the topology of the tree without any branch length information. So just keep that in mind. If you have that set, it'll look quite different than our rectangular phylogenetic tree here. There's also midpoint root, as I mentioned. So it roots the tree at the midpoint of the longest points between the two tips. So depending on what you're looking for, and changing the root position, changing the branch length, just use caution because this can drastically change the topology of the tree. And you'd want to make sure you're using an appropriate method based on your data. For today's workshop, we are just going to use the defaults, which is rectangular, and we're not going to be rooting it at any specific point or position. Is everyone able to make it up to this point? Yeah, so identifying nodes is pretty important to help understand the structure of the tree. So this is displaying the internal nodes. So we do that using geomtext2 onto our ggtree. And we subset it to not include the tips. So it just includes the internal node labels. And then it horizontal adjusts it a little bit so it's not overlaying right on top of the tree so we can read it a little bit better. And by doing this, we can utilize the node numbers if you want to, for example, zoom into this node. We know, okay, let's zoom into node 17, or let's zoom into node 27 just to get a little bit honed in view of a tree. It's good to look at the different nodes for that. There's other different tree manipulation options. Like I mentioned, you can zoom in to view a particular clade, and you can do this using view clade. So there is actually a difference, which I didn't know last year when I was teaching a similar workshop, between the plus and the pipe function. So this pipe function with the percent symbols, if you pipe your ggtree into view clade, then it'll just show the zoomed in clade. And then we're zooming into node 17 here, which is the long cluster. But if you do plus, it'll actually show both the original tree and the zoomed in clade, which is quite interesting. So depending if you wanted to show both in one visualization, that could be helpful. Other options are to scale a clade. So for here, I scaled, this is the original tree on the far left with the nodes, and I scaled by the node 27, which is right in here. Anything greater than 1, any scale that's greater than 1, it'll make the clade bigger. And if we wanted to shrink a clade down, we would do less than 1, like 0.5, 0.1, or something like that, if you wanted to highlight a particular part of the tree. There's also collapse. So we can collapse at a specific node. For example, this long branch, if it's kind of redundant information or we just wanted to collapse it for something, you can collapse it and then it kind of expands the rest of the tree. Did everyone have a chance to play around with those? Yeah, and there's different tree manipulation options. They're really fun and interesting to play around with just to see what they do. So instead of just having the lines, you can add tip labels. So you can check the name of your tip labels by typing in, by running the code head. So if you check head of the tree and with the $ tip label, it should pull up what is showing as your tree tip labels. So for us, it's the SRR numbers. And so if we add tip labels using Geom Tip Lab again, we're going to set the size to 4. And then there's these other 2 arguments I'd like to add. One is chord Cartesian, I think that's how it's pronounced, clip off. And this allows us to kind of draw outside the plot. If not, sometimes your tree will get cut off just by the R plotting system. Another one is theme, and this kind of using plot margin. And this creates a margin again around the plot, so it helps it not get cut off. It keeps some space around the plot. But we're focused here on Geom Tip Lab. So if you run that, then you should be able to see, oh, I guess it's the SAM numbers, not the SRR numbers, but you should be able to see the default tip labels. So it tells us a little bit more about our tree. You can also customize your tip labels. So sometimes you want to show information that's maybe a little bit more helpful, or if your tip labels are maybe private institutional numbers or codes and you don't want to show those, you want to just have them labeled as sample 1, sample 2, for example. You can customize the tip labels to be something else. So since we will be linking the metadata file to the tree, we can use a vector first to check if there are any SAM-N numbers, which are the biosample IDs, that aren't matching the tip labels of the tree file. So you want to make sure the metadata file matches the tree file. And the way we do this is by running this code and subsetting it to see if there's any tree tip labels that are in the metadata sample. So if you run that, you should get a character 0, meaning that all observations match between the tree and the metadata file. The important thing with this is that your metadata first column should match the tree tip labels when you're linking them. And then you can link the metadata and change the tip label using Geom Tip Lab. So run our original ggtree. And then there is this operator that's used to attach annotation data to the tree. So we're going to attach our metadata because that's what we named our metadata object. And then we're going to have the plus symbol and we're going to do genome tip lab. But this time we're going to add the aesthetic and label as sample ID. The default was biosample ID. And then the same chord Cartesian and theme. So when we run that, now we should have the samples. So we're going to run through an example here. And this is going to be the end product that we're going to work up to. I kind of made it look similar to the paper and the tree that was in there, but couldn't get it exact based off of just using a subset of data. But we're going to work our way through and add the elements one by one. So I'm going to go to my R. Okay, let's scroll down. Does anyone have any questions up to this point? Okay, so the first thing we're going to do is we're going to actually save our simple standard ggtree here as an object and name it, I'm going to name it ggsimple. So you can run this and it's linked to the metadata already. And it has these standard plotting functions. So we can run that. And if we want to view it, you can also just type it in here. And let's view it. Let's see what it looks like. So yeah, it looks like what we saw before, which is great. But now it's actually saved as an object. So we don't have to keep typing out this whole script anymore. We can just refer to ggsimple. So I'm going to use a tree manipulation argument called flip. And what this is going to do is reorient the tree, and it's going to flip node 25 with node 17. Remember, node 17 is part of this long cluster, and 25 I think is these guys. And the reason I'm doing that is just based off of the journal's tree topology. So that's why I did it, just because it was positioned a little bit different than what we have. So when you run that, then it's flipped a bit. Next thing I'm going to do is add the tip labels. So at this point, I'm not going to save the tree until we get to the final point. So building off of the ggflip, we can add the tip labels, and I'm going to make sure I offset it a bit from the tip points. Sometimes it's nice to have a little bit of space between where the points are going to be and your tip label so you can read it a bit better. Now we're going to add the tip points onto ggflip, the same tip labs as before, and we're going to add geom tip point. And we're going to add two aesthetics. We're going to add color, and we're going to color by looking at the metadata file. We're going to color by the different gene options. And then we're going to have the shape be matrix. And their reference to matrix is basically sample source. We're going to have size B4 and alpha 0.7. And I'll show you what that means in a second. So the alpha makes it a little transparent. If you have the alpha set to 1, I think it's completely solid. And then the less you go, like 0.2, it's a little bit more transparent. And sometimes it's just nice to have it that way. for aesthetics. But so now that we did that, we can see, okay, oysters, stool, water. And then the different colors for having both genes negative, one positive, negative, and then both of them positive. The next thing we're going to do, it's not too different from how the journal looks itself in the figure. But if you wanted to, for example, change the order of these and have maybe like stool be circle or water be triangle, you can specify some of these things using the scale color manual options. So if we do scale color manual, that'll help us specify which colors we want to be. For example, if I want the positive to be deep sky blue instead of the default blue that it is, or if I want a medium turquoise or a coral, these are all different color options that are under, if you look up ggplot2 colors, they have loads of different colors by name, or you can use hex codes. You can even just try and type in pink, purple, usually something will come up. And then there's also scale shape manual. So these values, they were pretty much the same, but they have numbers such as 16, 17, 15. And if we go, just going to go back to here, you can look up these codes as well. And they have, these ones are filled in. You can also have ones that are just outlines or other different shapes. So that's what the numbers orient to. Okay. Next, we're going to add a CLADE label. So that's going to add a label onto this long cluster here, just to show that that is sequence type 36 that they were highlighting. So everything is kept the same as before. Oh, sorry, I didn't run this one. Let's run this. And another thing that the scale can do for shape is that I specified a name. So what this does is it changes the name of the legend. This one before was just matrix, and instead I wanted it to say sample matrix. So the CLADE label that we're adding next is Geome CLADE Lab. And we're going to add it to node 17, which is this long cluster. And the label we're going to add is going to be in quotes, and it's going to say ST36 for sequence type 36. We're going to offset it a little bit. You have to play with offset quite a bit. It really depends on the size of your tree, how much to offset. And then we're going to set the bar size. I think that's the thickness of the bar. Bar color, I just selected gray 44, which is a little bit darker of a gray. Same with text color, and then text offset, whereas bar offset is up here. So let's run that. Oh, so as you can see, it's not super. It's just because this panel is a little bit small, but you can kind of get the point here that the offset, sometimes you have to play around with it a bit. Move that over. And then the last thing we're going to do is we're going to customize the legend a bit by adding in a theme. So there's so many different theme options you can do. You can change the legend text size. You can we're going to make it bold here. The legend title as well. So that is like the sample matrix, whereas this is the legend text. You can change the spacing. I just have that as 0 right now. You can change the vertical spacing and the horizontal spacing. You can make a border around the figure itself, and you can also make a legend border and change the color of the background if you really want. So we're going to run this and see what it looks like. Okay, Looks a little weird just because of the panel that I have, but if you open up and zoom, it might look better. Yeah, so that looks a little bit better. And to export the final tree, this is just kind of the basic way to export it using ggSave. And we're going to point it to our output folder that we created. You can give it any name you want. We're going to make it a JPEG, but it can hold a lot of different extensions, TIFFs, PNG, and we're going to save this example 1. And I set the DPI to 300. But there's also different options like specifying height and width. But when you save it this standard option without adding height and width, it'll save it the way that you view it right in this window. So if I saved it, this line would be kind of in the middle. So you might want to move it a bit so it looks a little bit nicer, and then you can save it. Was everyone able to follow along with that example? Okay. Let's go to notes. It's also in here if you want to look at that. So we're going to apply some of the things we learned already so far today to a little exercise. So what I want you guys to do is to create a tree, this time using Surovar as the tip labels instead of the sample IDs that we had. Then you're going to set your own color to the nodes based on sequence type. And you can also change their shape. You can change the font size of the tip labels and the legend if you'd like. And then a little bonus is you can move the legend to the bottom of the tree. If you're curious on doing that, you can look at the help page under theme, or you can Google just how do I move a legend in R, and it'll help you. I'll give you guys about 10 to 15 minutes. This is kind of what it should look like. Obviously, you can use whatever colors you want instead of trying to fish for these exact colors. And I can leave this page up here as well. Another hint down here, which is pretty important, is that when you're dealing with a continuous variable, such as numbers, where it's could be 1, 2, 3, such as sequence types or continuous variables, you're going to want to use as.factor and put the sequence type in there. If you're having issues, you can try and color by a different variable as well and see what that looks like. And if you need to look up any colors, if you want to customize them, you can search for ggplot colors. And the codes for the shapes are a couple slides back, or you can just try and type in a random number and see what comes up. So I'll give you guys about 10 minutes. Oops. Sorry, I'm reading direct messages. I can't see them. I'm sorry, I don't understand this question.
I think it's like you want to change the file name, should they run a code and then make sure the things change and then change the legend name?
Yeah, that's kind of why I do it step by step, because I like to see if you do just change one thing at a time, it's easy to see what it changes. But if you kind of run too much at a time until you get kind of familiar with what each argument does, it's nice to run it one at a time. Otherwise, you can get confused to see what each argument changes. Yeah. Okay, it's been about 10 minutes, and just for the sake of time, were people able to create at least some sort of variation? Ah, good job, Grace. Nice. I know it can be difficult when it's something that we just learned and I'm putting you on the spot, but this can be a really great way to try and learn is just by practicing. But the code is there in the answers script for you to look at, but I'll go through it quickly just so you can have a look at what I did. So what was a little bit different with this tree was that I used ggflip, so that already has the metadata linked to it. And that way I was able to customize the geome tip lab to specify the aesthetic as label equals Cerebar. And that's how I got the tip labels to do that. I upped the size a little bit to 5, which is a little bit bigger than the default. For geome tip point, I used the aesthetic and set the color to sequence type. So as I was mentioning, sequence type is a continuous variable. So without running as.factor, it might not work. So sometimes you have to do that when you have continuous variables for it to kind of read it more as a discrete kind of categories. And I set the shape to 18. I think I made the size a little bit bigger, and I made it a little bit more transparent by setting the alpha to 0.5. I just set arbitrary colors using scale color manual, pink, blue, green, etc. And I named the legend sequence type. For the theme, I made the font size a little bit bigger. I moved the legend to the bottom by doing legend dot position. So the default will usually, I think, be on the right-hand side, but you can add it to the top, you can add it to the side, you can flip it horizontal. There's so many different ways you can change the legend. You can overlay it on top of the figure as well. I set the legend background to blank. Yeah, so if you guys want to try running that as well, it'll be on the answers script. But just going to show you a few more things before we finish. So there are tons of different arguments you can try. Another one that's pretty popular that I've used is adding different layers for tree annotation. So aside from just adding one tip label, you can add multiple tip labels. So building off of what we did in exercise 1, if you guys haven't run exercise 1, or if you want to try and running it off of example 1, you can do that as well. But basically, you can add a second geome tip lab. It's based off of what's saved already. And same thing, add an aesthetic. And this time I'm going to make the label matrix. Offset it a little bit more because otherwise it'll sit right on top of the other label that we have going. I'm going to change the color to gray. And you can keep continuing to add layers this way while changing the offset each time, which can get a little tedious, but I continued to do that about 3 times. And so I got something that looks like this. We've got the original tree. I added the sample matrix. I added the year. I added the biosample number. And obviously this might not always be as practical for visualizing a tree, but for demonstration purposes, it kind of shows what we need to show. Another popular argument is highlighting clades. So there's a couple different ways you can do it. You can highlight by a specific node using geome highlight. You can select that you want to highlight the nodes 11 and 12. You can fill it. So this will highlight it by a certain color. I selected pink and I had it be alpha just because for highlighting it can be kind of dark. And then I extended it past a certain point. If you change this, you can see how it, the extension changes, but, and you can also highlight it by subsetting by a condition. So in this option, I did genome highlight and I mapped it to a certain aesthetic and I subsetted it based on, in our metadata, there's a column that's whole genome cluster. And I said subset and highlight whole genome cluster that is 1. So cluster 1, we're going to highlight as pink, and then everything else was the same. So that comes out with pretty much the same answers when you do that. And then I highlighted based off of the other clusters. So this is cluster 1. I highlighted cluster 2 as blue. And this other cluster 3 that's on its own, I highlighted purple. And the extend will just extend it a little bit past. So I have it covering the entire label. Just a couple more examples that I'll show you, but we're not going to work through. The capabilities of ggtree are really awesome, although it can be frustrating at sometimes. Once you get going with it and practicing, it's nice to have a reproducible tree. So this one here is an example that's in the ggtree book, and it's an example of plotting SNP data along with your phylogenetic tree and some different traits. So sometimes the trees can get pretty complex, and it kind of faceted the tree by tree, SNP, and trait. Here's another one that implements gheatmap, which is another package that complements ggtree and ggplot. And this is a tree that I actually built last year, perhaps, based off of this paper here. And I basically did the same thing we kind of did in this workshop, where I downloaded the genomes, I assembled them, and I tried to recreate the figure that was in the manuscript. And it's a really great way to learn. And if you're interested in learning gheatmap, there's actually a workshop that should be available from last year, where we kind of made a similar, more simple heat map. This is another example of a phylogenetic tree with using microbiome data and the abundance. And there are so many different resources available for ggtree. There's the book, which it has maybe 10, 12 chapters. It kind of walks you through everything from how to build a tree, what the different tree formats are, all the way down to the more kind of complicated visualizations. There's the ggtree GitHub. There's many different blogs and stuff available. People will show you how to do enhanced annotation using ggtree. And then something that came out very recently is ggtree Extra. I've only looked at it briefly, but it's kind of a newer, it is a separate package that kind of extends upon ggtree. So just a reminder for next week, if you'd like to bring your own data set and come in person, please sign up. That way we can get numbers. And if not enough people sign up, then we'll just do it virtual, but definitely let us know so we can plan a little bit for that. And does anyone have any questions for today? That was it. Yes, please make sure you fill out the exit survey for today. We would really, really appreciate it and help us improve future workshops.